All right, so this chapter is about DNA. So let's start off with a little bit of history. Frederick Miescher studied salmon sperm and pus nuclei. Pus is actually a substance composed of cell debris, infectious material, white blood cells, specifically neutrophils that are recruited to sites of infection. And what he did was he actually extracted the white blood cells of pus and looked at their nuclei to study heritable material or genetic material. He concluded that this genetic material, whatever it was, contained too much phosphorus to be protein. So he kind of ruled out protein as a macromolecule that could be composing this genetic material. And he called this heritable material nuclein, and that was later changed to nucleic acids, or nucleic acid as the macromolecule. Now we had Phoebus Levene. Levene, he was a skater boy. He figured out, or he discovered the parts of genetic material, or the anatomy of the genetic material. And by the anatomy, I mean the molecular components. So DNA is going to be composed of sugar, whether it's ribose or deoxyribose, ribose in the case of RNA and deoxyribose in the case of DNA. It's going to be composed of sugar, phosphate, and then one of four different bases. So that's kind of what he figured out, the kind of the formula of what makes up a nucleotide. So he coined the term nucleotide. Nucleotide is something that has a sugar, a phosphate, and then one of four nitrogenous bases. And so his first idea, or his idea about the structure of this genetic material, was a tetranucleotide, and that structure would look like this: tetra meaning four, so four nucleotides all bound together. So DNA has four bases with equal ratios of each base. So that was Phoebus Levene's idea. Now comes along Erwin Chargaff. So he was an Austrian biochemist that worked in the US, and his important contribution was that he found the ratio of your nitrogenous bases to be regular. Meaning, if you were to take a sample of DNA and extract all of the guanine, the amount of guanine that you would have within that sample would equal the amount of cytosine that you would have in that sample. So guanine, the amount of guanine equals the amount of cytosine. Likewise, the amount of adenine that you'd find in a sample would equal the amount of thymine in a sample. And then he also found that the composition, or the ATCG ratio, could be different potentially based off of the type of organism. He also found that there were two different purines, adenine and guanine, and then two pyrimidines, cytosine and thymine, in DNA. So we know that thymine will bind to adenine and cytosine will bind to guanine. So the amount of cytosine that you would have inside of a sample of DNA would equal the amount of guanine. So his main contribution is the ratio. All right, so now we have Linus Pauling. He discovered that proteins could form a helical shape, and he wanted to figure out the structure of DNA. He proposed a triple helix for DNA structure. Now we have Alfred Hershey and Martha Chase. What they like to do is study bacteriophages, specifically the T2 bacteriophage. And what bacteriophages have a tendency to do is infect bacteria. So it's a type of virus that will infect bacteria. So it's a virus that infects specifically E. coli. And so the anatomy of this is you've got DNA inside of this little capsule composed of protein. So DNA, or that nucleic acid material, which no one had discovered the macromolecule for it at the time, you have that nucleic acid encapsulated by a protein capsule. So here's the life cycle of a T2 bacteriophage. You've got this little squiggly blue that is actually the DNA, and then on the outside is going to be that protein capsule. And so what this virus will do is it will swim along in extracellular solution and make contact with a cell, and that cell being a prokaryote or a bacteria, it will inject its genetic material, whatever it is. At the time, we didn't know what composed it. It could be proteins, it could be carbohydrates, it could be, nucleic acids. So it would inject its genetic material into the bacteria, and then that bacteria would then take up that viral DNA and integrate it into its own genome. And then that viral DNA would usually code for enzymes and or proteins that would aid in the replication of the virus. So what it would essentially do is hijack the machinery of that bacteria to replicate more of itself. And then eventually when the virus reaches, some threshold, it enters this lytic phase, which would cause the bacteria cell to die, burst open, releasing many more viruses that can then infect further, more bacteria. So their main question was, what kind of compound or what kind of macromolecule is this genetic material? All right. And so they actually ran two different experiments. Both of them are going to involve radioactive elements. So let's look at their first experiment. What they're going to do is they're going to grow this bacteriophage in a medium containing radioactive phosphorus. So radioactive phosphorus. Now, a little fun fact is phosphorus is an element that is going to be found in DNA, but not in proteins. Okay? So they grow this virus in this solution, medium, rich in radioactive phosphorus. And so that phosphorus is then integrated into, all of the components where phosphorus would be integrated into, namely DNA. And so then what they do is they introduce it to bacteria. That virus infects the bacteria. Then they blend that bacteria viral solution in a blender, which would detach the viruses. So the viruses will infect the bacteria with its genetic material. That genetic material is then integrated into the bacterial genome. They blend it, separating the viral component from the bacterial component. And then what they do is they centrifuge that solution. And what a centrifuge is essentially what it does is it spins something around at such a high speed that it separates all of the very heavy components of a solution and the lighter components of a solution. And what you get typically at the bottom of a little Eppendorf tube is a pellet where all the densest, heaviest materials are. And so the bacteria actually will, all of the bacterial components will be contained within that pellet, and all of the viral components will be contained in the aqueous solution above, or the supernatant, what we would call as the supernatant. And so after they centrifuged it, what they did was they found that within the pellet, the bacterial components, that's where they found all of that radioactive phosphorus. And then in the supernatant, there was no radioactive phosphorus, and all of the, you could say, the viral component in the supernatant, there was no radioactive phosphorus. Now, with the second experiment they ran, is they had a medium containing a bunch of radioactive sulfur. So sulfur is going to be an element that you're only going to find in proteins, and you're not going to find that in DNA. So your virus is going to take up all of the radioactive sulfur within that medium, integrate it into whatever the sulfur would be integrated into, namely proteins. And then what you're going to do is you're going to combine that virus with bacteria. That virus will infect the bacteria with whatever genetic material it's infecting it with, and that bacteria will integrate that genetic material into its own genome. Then what they do is they mix it up in the blender, separating the viral component and the bacterial component, centrifuge it, bringing all that bacterial substance down to the bottom into the pellet, and then look at the results. And what they found was that all of the radioactive sulfur stayed in the supernatant fluid above the pellet in the viral component, implying that, oh, okay, so this sulfur is part of protein, and protein did not get integrated into the bacteria. So here's just kind of a recap here. Radioactive sulfur found in protein stayed in the supernatant, And supernatant is just going to be the liquid above the solid residue after, in this case, centrifugation. Now, only the radioactive phosphorus, which we're only going to find in DNA, is what actually entered the bacterial cells, because that was what was found in the pellet. Remember, the radioactively labeled stuff started off in the virus. The virus injected the bacteria with some substance, and what they were trying to do is figure out which macromolecule would actually be integrated into the bacterial genome. What was integrated into it was the nucleic acid. And so they were able to conclude that that DNA and not protein is actually hereditary material. So they were able to rule out through an experiment that proteins are not genetic material, or that genetic material is not composed of proteins. All right, so next we have Rosalind Franklin and Maurice Wilkins. Rosalind Franklin was a postdoc in the Wilkins lab, and both of them studied X-ray crystallography, which is essentially just crystallizing some substance and then using X-ray cameras to take pictures of them. So they crystallize things and they take pictures of very small things, down to where they can actually show the distance between atoms. So what they found was that DNA, or genetic material, showed a helical pattern. And these images were actually taken by Rosalind Franklin's, one of her graduate students. Okay. Now we have Francis Crick and James Watson. I believe Francis Crick had his master's and Watson had his PhD. And they were both working in the Cavendish Laboratory at Cambridge. And so they communicated with Wilkins about Franklin's photos. They knew about Linus Pauling's models, and they used Chargaff's ratios that he discovered. And what they were able to do was take all of that data and deduce a model of DNA using all that data. So they didn't actually run any wet experiments necessarily. What they did was they just took all the data from all the different labs and mashed it together and came out with a good structure of DNA, or they discovered the structure of DNA. Now, there was a lot of controversy around this because Franklin did contribute her images to the deduction process, and so it's thought that maybe she should have received some kind of credit for the Nobel Prize, because James Watson and Francis Crick eventually, because of their discovery of the structure of DNA, won the Nobel Prize. Okay. So we've got a few features of DNA. Here's the first one, is that it is a double-stranded helix. So it's double-stranded, meaning you have one strand and it's connected in the center to another strand. So it's two-stranded. And we have this sugar phosphate backbone, which should look familiar. You got the phosphate here, bound to a sugar, bound to a phosphate, bound to a sugar, bound to a phosphate, bound to a sugar, and so on. And then you have these nitrogenous bases that dangle off in the center, and they actually make attachments to the nitrogenous bases on the opposing side through hydrogen bonds. So they make hydrogen bonds in the center. Your A, or your adenine and thymine, those will make two hydrogen bonds. That's what links them together. Two hydrogen bonds link them together. And your guanine and cytosine actually have three hydrogen bonds that link them together. The way I like to remember this is that apples will typically fall off of trees pretty easily because they're only held together by two hydrogen bonds. And girls, they have a high amount of hydrogen bonds with their cats. So they have three hydrogen bonds. All right. Some other fun facts is that your DNA is going to be uniform, or going to have uniform spacing, and also that it has a right-handed twist. Meaning, if you were to look at the DNA from above, it would actually rotate in a clockwise fashion. Another way to remember this is if you hold out your own right hand. I don't know how this would work here, but if you hold out your own right hand and hold up your thumb, so I guess it would be like this, and you twist your hand in the upwards direction, that's the way it would twist. That's the way DNA would twist. All right. And then we have this other fun fact is that the two strands, yeah, it's double-stranded, but they actually run anti-parallel. So what do we mean by anti-parallel? Well, it's anti meaning not, and parallel meaning same, right? Not same. Opposite directions. Anti is opposite. And so if you look here, you have a 5 in this little dash here. That means 5 prime. So you have a 5 prime end, and that runs in this way, and it ends in a 3 prime end. And then over here, you have a 5 prime end, and it runs this way until a 3 prime end. So what does that actually mean? So in organic chemistry, when naming things in organic chemistry, what we have a tendency to do is name or number the carbons in that organic molecule. So let's take this molecule, for example, or this nucleotide, for example, and you have your first carbon here. So carbon number 1, or your 1 prime carbon, your 2 prime carbon, your 3 prime carbon, 4 prime carbon, and your 5 prime carbon. This 5 prime carbon is bound to a phosphate, and this 3 prime carbon is actually bound to a hydroxyl group. Okay, so if we're starting here, you have this 3 prime carbon bound to the hydroxyl group, and then you have this 5 prime carbon bound to this phosphate, which is bound to the 3 prime carbon, which is bound to this, this 3 prime carbon, which is bound to the 4 prime, which is bound to the 5 prime, which is bound to this phosphate. So it actually runs in this direction, from 3 to 5 here, and then in the opposite direction, you have the 3 prime carbon bound to the hydroxyl, and then your 5 prime carbon bound to the phosphate, and all the way down, 3 to 5, 3 to 5, 3 to 5. So that's what we mean by anti-parallel. So carbons are given a prime number, or given prime numbers in deoxyribose. Strand runs 5 to 3, and they link up to the strand running in the opposite direction.